it doesn't come forward? Okay. So briefly speaking, stem cells are the cells that are capable of self-renewal and can be induced to different lines of cells. At the beginning, we have a symmetric cell division when we are at the level of 8 cells, the first 3 days of development of an embryo. It means that each of those cells are identical to the other one, and they are capable of producing any tissue. Then we will have a little bit of asymmetric cell division. So the cells, the new cells are somewhat different, and that green cell identify the cell that may be generating a new tissue, but not all of the tissues. So let's look at how does it happen. First, we have a fertilized egg. Then for the first 3 days, we have totipotent stem cell. What totipotent means is this. They can produce any cells in the body. In addition to that, they can produce placenta. The next stage is when we have pluripotent stem cells, and that's from day 5 to 15. Those are the cells that can produce any cells in adult human, but they cannot produce the placenta. And after day 15, the cells differentiate more, so we have stem cells, but that the stem cells are directed toward, for example, generating blood or generating some specific tissues. So let's identify and distinguish between totipotent, which means that they can produce placenta. Pluripotent means that they can produce any cells in the body, and then the lineage. And then we have adult stem cells, and they are producing some specific tissue throughout the adult life. So we are all familiar with limbal stem cells that produce the corneal epithelial cells. And then we have some latent stem cells in the eye, like RPE latent stem cells, ciliary epithelium that can produce some of the cells in the eye, and Muller glia. That's one line of the stem cell that is generated from the embryo. But then there is another line which is induced pluripotent stem cells. That's based on the Nobel Prize work of Gordon and Nakamoto in 2012. They got Nobel Prize because they showed this. If we transfer 4 genes in an adult, we can actually induce the stem cells. For example, if a person has a disease, an adult, you can get a muscle cell and change those 4 genes, that become the specific stem cell of this individual, this person. And from those stem cells, you can generate the tissue that you need, for example, neurons or other things. It's not as simple as this diagram, but that can be done. So basically, we have 2 lines for stem cells. Then we have embryonic stem cells that come from the fetus embryo from the first 2 weeks of life. And then we have induced pluripotent that we can get from adults. When we look at the stem cell transplantation in retinal diseases, there are multiple lines or ways that we can do. One is injecting RPE cell suspension. That's just injecting under a bleb with some cells. That seems to be the more easier way of doing that. And then a little bit more complicated is when we actually transplant the sheets of cell instead of those individual cells. On the other hand, when we want to go and transplant retinal progenitor cell, that's much more difficult. Why? Because we need to have neuronal connection. So we need to have actually connect axon and dendrites in the retina. It's not only enough to have some cells surviving, but you need them to function. And a little bit more complicated is when RPE sheets and retinal progenitor sheets are transplanted in a sandwich. And that would take a little bit of time until we get to a successful level 5 of this diagram. Currently, there are 37 clinical trials involving stem cells in ophthalmology. One of them that Dr. Grigori just mentioned was a paper that was published in Lancet. Dr. Del Priore is an author, and it was 9 Stargardt, 9 dry AMD, and vision improved in 10 eyes, same in 7 eyes, and worse in 1 eye. For example, here, when the stem cells was injected in this geographic atrophy, in this area of the circle, then slowly the RPE progressed. This is RPE cell suspension transplantation. Currently in our lab, we are working on some new ways of doing the stem cells. One of the things is when you have a stem cell, for example, RPE stem cell, when it develops, it's not as normal as when the RPE and retinal progenitor cells are next to each other. Because when you have a co-culture, the cells actually send message to each other. For maturation of the retinal progenitor cells, we need RPE cells. And for maturation of RPE, we need retinal. The two need to work together to have a successful cell. And that's where the co-culture comes from. So we can actually transplant those co-cultures, use them in a scaffold, and then that can be transplanted in the retinitis pigmentosa mouse model by injecting them under the retina. This is an example of what happens in RD10 mice, that more outer nuclear layer is preserved in this model. And also, we have tried that in pig models, transplanting the sheets of cells under the retina. So in summary, while clinical trials have been encouraging, this is a still very experimental treatment. Human stem cells can replace defective ocular tissue. We are still at the beginning of this, and it will become better and better. A major challenge is to restore the inner neural network connecting axon and dendrites. And another use of stem cells is that when we have a dish of organs of retina, then we can actually study the medications for safety in that dish. Thank you very much.